I want to talk to you folks about work that a Ph.D. student did in the lab, John Pribis, and you can see him here with collaborations with many other labs and lots of people in the lab. Most notably, you'll hear some new unpublished work of Yin Zhai, who's next to John here. And John has moved on and is in Boston now. So the story I'm going to tell you is this cool thing that John discovered, that when bacterial cells are exposed to ciprofloxacin, one of the most commonly used antibiotics, they induce a pathway that produces mutations. And luckily for us, it is a pathway that we had actually figured out over many years of work, not with antibiotics, but with starvation. And he discovered that what they do is produce a little subpopulation of what we call gambler cells, and that these cells specifically do the mutagenesis. All the rest of the cells in the population kind of just hang out and don't hurt their genomes. So it's like these ones are rolling the dice. And if they do the wrong thing, there's still some cells that didn't mutate. And if they make mutations that confer resistance, well, then it was worthwhile for them to do that. So I'll tell you a little bit about this mutagenesis mechanism today and how these gambler cells come to exist. And you'll see here, the take home is that the antibiotic induces reactive oxygen that causes upregulation of a couple of small RNAs that allow the translation of a stress response activator, sigma S, which activates the general stress response, and that this allows a pathway of mutagenic repair of double-strand breaks, which we call mutagenic break repair. Oh, and then the other super excitement about this is it has been our dream for many years that if you could understand the actual molecular mechanisms of how evolution works, you might be able to make drugs to inhibit it. And what I mean by that is, if you imagine giving an antibiotic, and that antibiotic is not just helping a patient recover from a pathogen, but at the same time, it's spurring evolution that could lead to resistance, it would be great to have a drug that you could give that would let the antibiotic do its job without generating resistant mutants, which are just, a major problem in the modern world. So we discovered one, John did, by his smarts, you'll see. All right, so to get you into this antibiotic problem, I guess we have to think about mutagenesis as a whole. And actually, before I say that, I should say, in this group, and please forgive my laryngitis. In this group, you know that you can get resistance to many antibiotics, both by horizontal transmission of genes, but also by mutations of existing genes in a bacterium that will give it resistance. And although the horizontal transmission part is really very well studied, the mutagenesis part, which is really important clinically, we knew very little about. And our interest started in really wanting to understand how evolution works. What you learned probably in school, and what I definitely learned in school, was kind of like this, that you have populations of individuals who have genetic differences between them, this heterogeneity. They go into a selective environment, and in that environment, whoever has the right stuff wins. And that has been a view since the 30s and 40s, and for good reasons, but you're going to see that it's really not what's going on. I'm going to tell you that instead of that, when cells are put into an environment that's selective, and evolutionary biologists would say, but we molecular biologists would say it's stressing, that environment will activate stress responses that you all know about. And that in addition to protecting the hardware of the cells, these stress responses turn on mutagenesis programs that increase the ability of cells to evolve. And we discovered this by really trying to get into the molecular mechanisms of how the mutations happen. So a summary of previous work is that we learned that mutagenesis is regulated temporally by stress responses. And they do this very cool thing of making it so that a cell that's poorly adapted to its environment, one that feels stress, is more likely to generate the mutations including mutations that will let it evolve. So that's really interesting and different from the neo-Darwinist view. And then we also discovered that mutagenesis is localized in genomic space. It happens in particular regions. Actually, we heard this from Kathy about RNA, and it happens in clusters. And that's actually important for evolution too, because a big problem is for people who care about protein function is how do you change the function of one protein to another? Almost always, it is never a single amino acid substitution. It almost always is multiple. And that problem can be addressed. And people worried about that. What's the chance of you getting multiple? But if they actually come together, you could see that that would be potentially evolution promoting. So these things that we discovered working in E. coli, happily over the last at least 10 years, these have become obviously going on in organisms all across the tree of life, bacteria, eukaryotes, humans. I'll show you a large gene network or a large protein network that promotes this. And the take home from that is almost all of it is devoted to turning on those stress responses, showing you how important that is. And then finally, we'll get into the limitation or regulation of mutations to mutagenesis to happening in a small cell subpopulation, which we'll call the gamblers, and that antibiotics induce this. Okay, and this is the take-home message for people who would like to go do something else now. When a cartoonist in Cambridge drew this when I was giving a talk there, and I just loved it. It's a bacterium complaining about feeling bad. And the other one says, time to flip your switch. And her idea was that it's like this. Turn on to speed up evolution, emergency use only. So the stress responses. We were studying mutations in E. coli in an assay that I won't show you, so I'm hiding it under this fig leaf. We can pretend that this is it. It's kind of like it, but not really, where wild-type cells in a stress environment make lots of mutants that you see as little blue papillae on a colony. And a mutant that lacks a stress response activator makes few. So that says that that stress response is required for mutagenesis. And doing experiments like this, we discovered that the general stress response is required for mutagenesis under starvation stress. So is the DNA damage response, the SOS response, and so is a particular membrane stress response regulated by sigma E. And for all of these, it means if you stop the response, the mutations don't form. We do a lot of controls that demonstrate its mutagenesis and not some other problem of the cells. So because of that, I think it's okay for us to call mechanisms like this stress-induced mutagenesis, their definition being upregulated by stress responses. Before that, we actually learned a bunch about the mechanism of the mutations in starving cells. The mutagenesis mechanism required the proteins that do double-strand break repair by recombination. My first graduate student's first paper, that the mutations look like DNA polymerase errors, which led us to think it was mutagenic repair of double-strand breaks, and that a special low-fidelity error-prone DNA polymerase is used. And Becky Ponder, when she was a genetics student a graduate student in the lab, did some lovely experiments that revealed the mechanism that I'll just tell you the take home from. We learned that how this mechanism works is that DNA will experience a double-strand break, and you can think of that as just spontaneously. There is some spontaneous frequency of this happening. When it does that, it'll activate the DNA damage response, and it in fact does that in most cells. So you get point number 2 for free. The SOS response transcriptionally upregulates this error-prone polymerase about tenfold, and believe it or not, even with that really in abundance, repair starts to happen, but it will not be mutagenic until a third thing happens, the activation of the starvation or general stress response by sigma S, which is activated by all kinds of things that are not DNA damage: starvation, cold shock, acid shock, osmotic shock, oxidative stress. And then, and only then, do this and a couple of other error-prone DNA polymerases get to leave their errors in the newly synthesized DNA in a repair reaction. So Parallel lines or strands of DNA, dotted lines are newly synthesized DNA, and the X means a polymerase error that becomes a mutation. This model that came from Becky's in vivo work, we were so happy to see that biochemically it holds up. Mike O'Donnell's lab showed, and we know a lot more about it. This is a review that would be helpful if you're interested more. So I want to tell you some experiments of Chen and Shi, a postdoc in the lab several years ago, where he got to illustrate this in a particularly nice way. He took E. coli cells that have an inducible restriction enzyme, a double-strand endonuclease, and they have engineered into their chromosome a single cut site next to a gene that doesn't function, but if it gets a frameshift mutation, it'll confer tetracycline resistance. And his experiment is so simple. He just grows the cells, let them starve, and then plates them on plates where they have food, but they also have tetracycline. So the frequency of TET-resistant mutants per total is that we use that to calculate mutation rate. These are frequencies here. What you can see is when he gives the double strand break, DSB, there's about 50 times more mutation than spontaneously when there's no break. This mutagenesis requires the sigma S response and the error-prone polymerase, DNA polymerase, DNB, and moreover, it happens over time during starvation, which makes sense because that's how the starvation stress response is induced. So this is just an illustration of one way you can study this, and other experiments I might or might not show you at that time point. This is a different experiment. This is actually Chandon did this, it's log phase cells, not stationary phase starving cells. And here, when cells are growing happily and they have plenty of food and they're not stressed, he can give a double strand break. It will induce the SOS response. But if we look at the frequency of TET-resistant colonies, there's no increase with double strand breaks compared to no double strand breaks. However, if he artificially produces just a tiny little bit of that transcription activator, sigma S, he gets mutagenesis, and of course it's sigma S and DNB dependent. This I wanted to show you because what it says is that it's not really the stress itself that makes this happen. You can take unstressed cells, it is the stress response, the sigma S regulon, which encodes hundreds of proteins. Okay, so stress is not necessary. The stress response is really what does it. And in experiments that he did, and Becky did some similar ones, he showed this amazing thing that you don't actually need to do this. You don't need to use sigma S or have this error-prone DNA polymerase to repair a double-strand break. And by that, I mean you can cut the DNA with a restriction enzyme if it's repaired while the cells live. And the frequency of them living is no higher if they have the error-prone polymerase. And in one experiment, it was actually lower. What that means is that cells are not doing this because they need to repair double-strand breaks. They can repair double-strand breaks very well without this. So what all of this means is that there's a switch that I don't want to say normally because I think stress might be kind of normal. In unstressed cells, there's double-strand break repair by recombination, and it's high fidelity, low error rate. But under stress, we get a switch caused by the activation of the sigma S response that allows mutagenic break repair. And all of this makes it that cells that are in an environment that they're poorly adapted to can actually increase their ability to evolve. I'm not showing you data that you may wonder, are the mutations near the double-strand break site? And the answer is yes, within about 100 kilobases around it. Let's take a very brief look at the network of proteins that supports this. Mary Jane Lombardo, when she was a postdoc, followed by Amar Al-Mamun, did a screen for, they did a transposon mutagenesis and screened for all kinds of mutations that would stop stress-inducible mutagenesis. And this list of 93 proteins is what they got. And the amazing part is that only a small fraction were the ones that we knew about, the DNA repair and mutation-related proteins. most of them were just other stuff that cells do, metabolism, electron transfer, proteases, various regulators. It was not mostly the DNA end of the deal. And doing a lot of experiments, this is kind of old and published, so I won't tell you them, but I'll tell you that of these 93 genes, more than half of them were leading to mutagenesis by virtue of activating a stress response. And for example, that means that if you don't have that gene function, but you artificially upregulate the stress response, mutagenesis happens again. So that was really all you needed it for. 30% of this network promotes the induction of the sigma S response. I think that says sigma S is pretty important to mutagenesis. The cell devotes a lot of proteins to it. And for the electron transfer genes, which were kind of mysterious to us at first, that's how they all work. They are about activating sigma S that makes the switch. So what this means, oh, and I didn't show you this because, as I said, this paper is old and published, but when we looked at the proteins that activate stress responses and the stress response activators, so like most of these proteins are feeling the environment and doing a whole lot of signal transduction that ultimately lets a stress response get turned on. It turns out that the stress response activators are actually the hubs of this network, which to me was exciting because it indicates that if we could therapeutically inhibit one of those hubs, maybe we could make the whole network fall apart and prevent stress-inducible mutagenesis, for example, in pathogens, but also in cancers, where others have shown that really the same kind of things happen. Okay, so now let's talk about ciprofloxacin. This is the work of John, and you'll notice that this is not John Purvis over here. It was because John, he was kind of a funny guy. I had to make a presentation before I had his photo. I put this guy in as a stand-in, and John was happy to be represented by him, so I'm leaving him in for the rest of the talk. So ciprofloxacin is a fluoroquinolone antibiotic, very, very highly prescribed. And resistance clinically occurs by mutations in about 3 or 4 genes. How quinolones, including ciprofloxacin, work is they're inhibitors of type 2 topoisomerases. These are the ones that break both strands of DNA, turn them around, and religate it. What these drugs do is they bind the topoisomerase after breakage, but before ligation. So it means that DNA breaks accumulate in cells. And it was already known that that induces the SOS response because DNA damage induces SOS. So Jim Collins's lab showed that quinolones and many other kinds of antibiotics induce a burst of reactive oxygen in cells, and that this is required for mutagenesis. And in this very nice paper of theirs, they showed, including making mutations that have nothing to do with quinolone resistance. So mutations that confer resistance to antibiotics that the cell has never even seen. We wondered, how did this work? So you can say, okay, you need reactive oxygen somehow to do mutagenesis, but how is it doing it? And I think, sort of the assumption was that reactive oxygen would oxidize DNA, and then you would get mutations from that. I'll show you that John showed this turned out not to be true. What's going on is reactive oxygen turns on the sigma S response and allows mutagenic break repair. So how he showed this was he developed an assay where he grows E. coli in low doses of ciprofloxacin that are called sub-inhibitory, a funny thing because 90% of the cells die, 10% of them live, but it's lower than a therapeutic dose. And then just growing them in a low dose of cipro and then plating them unselectively or on plates where he could select for base substitutions in the RNA polymerase gene that gives rifampicin resistance and any knockout mutation of a gene called AMP-D that gives ampicillin resistance. And what he saw was compared growing in cipro versus no drug, you get about a 25-fold increase in RIF resistance mutagenesis, about a 15 or so fold increase in AMP resistant. These ones, when are all different kinds because it's just loss of function. And they turn out to be mostly base substitutions and some deletions. And importantly, they are not mostly the signature that you get from oxidizing bases miscoding. So it didn't look like oxygen-induced mutations at the DNA level. The oxygen for John was required just like it was for Jim Collins' lab. If he puts in an oxygen quencher or prevents reactive oxygen in the first place, the mutagenesis goes down. This kind of way of showing it, this is going to be the frequency of mutants with drug, with cipro, over no cipro. So that's the fold induction of mutation rate. John did this very extensive series of experiments looking at the proteins that we already knew were functioning in starvation stress-induced mutagenesis. And he showed that it's an identical pathway as far as protein requirements go. It requires the stress response regulators for SOS and the general stress response, the proteins that do double-strand break repair. And this PAL4, I'm sorry, I have the name change, is DNB. It is the DNA polymerase error-prone that's most responsible. These guys, other ones, are for starvation minor players, and here they are more important players. And that's true for both kinds of mutations, so they are stress-inducible. I want to point out that the finding of mutagenesis upregulated by the general stress response, Ivan Matic's lab had showed that a couple of years previously for beta-lactams, which was a lovely paper. And it was interesting to see that the same thing is true for a very different kind of stressor, for fluoroquinolones. So if it's mutagenic break repair, making the mutations should require double-strand breaks that can be repaired. John showed that if he expressed in cells a protein that we engineered that binds double-strand break-ins but doesn't let them repair, when he induces that protein, GAM-GFP, the mutation rate of the rate of induction, cipro over no cipro, is reduced, which says that double-strand breaks are required for the mutagenesis. And here you can see them because this protein actually labels DNA double-strand breaks as foci. These are cells with double-strand breaks. You can see a focus here, that's no drug. And when you give the drug at this sub-inhibitory dose, you see plenty of double-strand breaks in these elongated SOS-inducing cells. And interestingly, like maybe your second favorite hypothesis for the oxygen is maybe oxygen breaks the DNA. Well, that turned out not to be true. Regardless of whether we quench the oxygen or not, it didn't change double-strand break levels. So now I want to show you the gambler subpopulation and that reactive oxygen makes it happen, and the drug that stops it. So John looked with flow cytometry at how many cells in the culture have reactive oxygen and how many have a sigma S response. And this is the reactive oxygen, it's with a dye. And for this, it's with a reporter gene that turns on with sigma S activity. And you can see that compared with no cipro in black, when he gives cipro, there's a lot more reactive oxygen, but there's a subpopulation with a whole bunch more. And then similarly, for the sigma S response, a subpopulation when he gives cipro, that says, you know, some cells are really inducing much more general stress response. And this general stress response is reactive oxygen dependent because it goes away with an oxygen quencher. It's about 20% of all the cells. And when John facts them, so he's now going to sort out the cells that are the high green ones that are left over that have low greenness, and compare with unsorted and mock. There are 400 times more mutants being produced by this little green population than there are by the low population, this big one. And that's why we call these guys the gamblers. They're a set of cells who are really turning up mutation rate. We know that they differentiate based on a sigma S response because we can sort them on that basis and find that that's where the mutants come from. And they're only mutable transiently. Once they get that mutation and they're no longer stressed, their mutation rate goes back down. This picture is just going to show you that if this is the experiment where you give ciprofloxacin, and now with flow cytometry, look at reactive oxygen and sigma S induction over time, sigma S on the bottom, reactive oxygen this way. Reactive oxygen comes first, and then the cells that induce a sigma S response, they are the cells that got high reactive oxygen. So it really is the case that the oxygen is giving a sigma S response. And I think, can I ask, is my time, am I out of time? Should I wind it up here?
You have less than 5 minutes.
Okay, I will just say a couple of more things and not tell you the news story. John, because he was really a buff and is on drugs and drug resistance, he knew that there was this FDA-approved drug called edaravone that reduces oxygen levels in people's blood. So this is a drug that can be prescribed right now. And he thought, if that's what it does, maybe it can stop mutagenesis. And in fact, that's what he saw. So here, and also these stress responses. Here's the sigma S reporter in flow cytometry. If he doesn't give the drug edaravone, just the vehicle that it goes in, here's your sigma S on subpopulation. If he gives it, you reduce this and lose most of those sigma S on cells. Same thing for reactive oxygen, the blue line here. And mutagenesis, importantly, is also reduced by edaravone. So this is super exciting because we have the first anti-evolvability drug, period, that I know of for any organism, including human cancer, where we're also interested. And it works by a mechanism that we understand, by suppressing the oxygen so the gamblers don't get differentiated. Okay, I want to leave enough time for you to ask me questions. We have figured out an initial differentiation event that I'm not going to show you, but you can ask if you're interested. And why don't I stop there? And yeah, we could stop here and take your questions. This is the pathway that John worked out.
Thanks so much, Dr. Rosenberg. I guess I'll get started with questions.
Please.
Could you tell us a little bit more about, do you know very much about the mechanism of edaravone?
Yeah, so edaravone is really an antioxidant. Okay. It's used almost not at all in the US. In Europe, it's used for patients who have just had a stroke or been in an accident so that they can preserve tissue while they're trying to rehabilitate the patient. And then in Japan, it's used as an anti-ALS drug. So ALS, Lou Gehrig's disease, a neurodegenerative disease. That one is pretty simple because it's just getting rid of this. And I think I showed you that it does. I didn't show you this, but John showed that it does not alter any of the upstream events like double-strand breakage, SOS induction. It really works here. We are so interested in finding more drugs to block evolution of pathogens. And in fact, we need to do drug screens. And I'm recruiting people to my lab now. So if any of you are going to be postdocs and want to do screens of molecules to find drugs to inhibit a pathway like this, we would love to go there. And what I was going to tell you is by an initial screen of an FDA-approved library, Yin here, Yin Shai has found a second anti-evolvability drug that I'm not showing you today. I am pretty sure that there will be many. And my dream is that not just to prevent antibiotics from causing resistance, including to some drug that the cell has never seen, but also maybe a drug that that slows down evolution of a pathogen. Maybe we could give this to people without giving them antibiotics. So if you think about that, when we give antibiotics, we are flushing our microbiome to a large extent. And we know that losing the microbiome, when it gets repopulated, it's almost never has as much diversity, and it's not good for people. What if we could just slow the evolution of pathogens so that the immune system could catch up without even giving drugs? Because the immune system really is doing an evolutionary arms race. So anyway, that's what I think will be in the future, the excitement of this, finding them, working them in preclinical models, and then actually doing clinical studies.
This is Steve Norris. I wonder if I could follow up on the edaravone mechanism. So does it actually decrease molecular oxygen levels, or just ROS levels?
ROS.
Right. That's true of antioxidants in general as well.
Yes, it is. Right. So that was why this one, it didn't come from a screen. John picked it because he thought, well, it ought to work. And if somebody wanted to, they could prescribe it tomorrow. So it was kind of a proof of concept drug. And, we're not under illusions. Before one goes to the clinic for clinical trials, you want to have a lot of candidates, not one or two. That's why I think we need to do a whole bunch more screening, because what my clinician friends tell me is that almost everything fails. So we'd like to have a lot of candidates.
But Susan, this is Heidi. So if this drug has been prescribed to patients already, has anybody noticed that those people are having, less resistance to infections or anything? Is there any data on that?
In the little bit that's out there, it's just not even examined. So there's a literature about slowing the progression of ALS. And then there's another literature about being able to preserve patients who have had a stroke for longer before they get some kind of treatment. But we haven't seen anything on studies of infection.
Okay, that might be interesting. Yeah.
Oh, you bet. You bet. Sadly, I can't really piggyback on somebody's US study where they're already using it, because it isn't used in the US. It's approved by the FDA, but it's not currently in a protocol for any particular disease here.
I have one other question, which was, so the gamblers are clearly different, but do you know the mechanism? Maybe I missed that, about why they have such a higher mutation rate?
Yeah. Well, I'll tell you what we knew when we published this, and then I'll give you a word about Yin's exciting unpublished results that I'd hoped to get to, but didn't want to use up my time. So we know from John and Yin's studies using flow cytometry to observe how many cells in a population are doing one or another thing. We built fluorescence reporters for both the SOS response and the sigma S response over here, and you can see reactive oxygen with dyes. What we knew is that when he follows the activation of these stress responses, most of the cells, not every single one, but really most of them have an SOS response. If we knock out their ability to make an S response, nobody gets reactive oxygen. So the SOS response is actually required for the reactive oxygen. Here's the mysterious part. Although almost all cells have an SOS response, only 20 to 30% of them get reactive oxygen. We knew, because John tested it, that also upstream of reactive oxygen is ubiquinone. So this is part of the electron transport chain, and believe it or not, cells are alive without it. If John looks at ubiquinone mutants, they don't make reactive oxygen either. So his hypothesis was that SOS was inhibiting the electron transport chain. When you do that, you get autooxidation of ubiquinone, and that releases reactive oxygen. This part was a hypothesis, whereas the rest of this picture is actually demonstrated. So the interesting work of Yin's was that, I don't know if I can go back just to show you this protein. Yin looked at a protein called TSB, and TSB is an SOS-induced toxin, like part of a toxin antitoxin system. It embeds itself in the membrane and disrupts the membrane. And it's known that when TSB is expressed, you have inhibition of electron transfer and growth. Yin has looked at it and shown that if you knock out TSB, which is not essential, you still get the SOS response, you don't get reactive oxygen. And you don't get reactive oxygen, sigma S response, or mutagenesis. So it's still like, for me, the fundamental mystery is, well, why are all the cells doing something that's required, but only to make the next step, reactive oxygen, but only 30% of them do it? Yin followed the expression of this protein TSB in E. coli, and unpublished, he has demonstrated that SOS induces a broad distribution of amounts of TSB in cells, and there is in fact about a 20% subpopulation that has a lot. So it seems like the fundamental event is the regulation of TSB, and the fact that it comes on in only some cells seems to be what gets this process going. So I'm very excited about Yin's work here because we have a real mechanism now. It's poking holes in the membrane, electron transfer goes down, reactive oxygen goes up.
Great, that's really, really cool. Thank you. Thanks
Thanks, Heidi.
for sharing that with us.
Nice to hear you, even though I don't see you.
Oh, here. I'm here.
Hey, Heidi. I don't know if anyone else had questions. Let's see. Oh, this is not stuff about my talk. It's about a raffle, so I'm not going to read the chat. Okay, so a whole bunch of students just graduated. People are moving on. Yin is about to this project. If you like this stuff, we are definitely moving forward, and I'm looking for people to do it. We're doing a bunch of other stuff too. I didn't talk about that today. Patricia, I don't know if you want to say anything else at the end of this?
No, just thank you for presenting your work, and it was really cool to hear about.
Great talk. Thank you so much.
And thank you, Kathy. I really loved your story. Thanks, everybody. Good to see you all, and bye.